This little lecture is about redox reactions in biology. So redox reactions in biology are either carried out by metals or by organic compounds. Biologically important metal ions are typically bound to proteins. They are not moving around freely in the cytoplasm. And such binding of the metal ion to a protein, such as the binding of iron to hemoglobin, can alter the free energy of the metal, its stability of the reduced form versus the oxidized form. Other than metals, organic compounds are also involved in redox reactions in the cell. And one of the most important of these is nicotinamide adenine dinucleotide, NAD+. NAD+ mediates many important redox reactions in biology. And in this half reaction, NAD+ is getting reduced by a proton and 2 electrons to produce NADH. NADH provides reducing power in the cell and is ultimately used to generate ATP in the mitochondria, as we will eventually see. But notice here that this half reaction involves 2 electrons. Some biological processes, such as photosynthesis, involve reactions with single electrons, and therefore we also have other types of organic molecules, such as flavins and quinones, which can also help with single electron processes. Okay, so how does NAD+ produce reducing power in the cell, and how is ATP produced from this reducing power? If you look at the oxidation of glucose, the complete oxidation of glucose has the following reaction. I'm not showing the physical state of individual components. And the free energy of this reaction under standard conditions is huge. It is about 2823 kilojoules per mole. So this is the energy that you would obtain if you burn 1 mole of glucose. So although there are no explicit electrons in this reaction, the oxidation of glucose can be written as a sum of 2 half reactions in the following way. Here we produce 24 protons and an equal number of electrons. And these can be then added to water to produce 12 molecules of liquid H2O. These electrons and protons are actually transferred to FAD+ and NAD+ to produce FADH2 and NADH2. So this process of glycolysis in the cell occurs in 3 steps. First, or glucose oxidation in the cell. The first is glycolysis, and you must have studied this in your first year biochemistry, followed by the citric acid cycle, also called TCA, followed by the respiratory chain. And it turns out most of the ATP is produced in the respiratory chain. In all, these 2 steps produce 10 molecules of NADH and 2 molecules of FADH2. So let us look at the energetics of these processes. So the oxidation of NAD+ can be written, or the reduction of NAD+ can be written in this way. NAD+ plus a proton plus 2 electrons gives us NADH. And the standard reduction potential, I'm using a prime to denote standard biochemical reduction. We will talk about it in the lecture separately, is this. And for oxidation, the reduction potential is like this, and here the standard reduction potential is 0.816 volts. Since the reduction potential of oxygen is much higher or more positive than the reduction potential of NAD+, so oxygen is a better oxidizing agent, so oxygen will get reduced, and the overall reaction is half O2 plus NADH plus proton produces NAD plus water, and all these are reversible, of course. So this is reaction 1 minus reaction 2, and the total cell potential for this will be equal to 0.816 minus 0.32, which is equal to about 1.136 volts. If you calculate the corresponding free energy of reaction, like we did earlier, minus the number of electrons, Faraday's constant, and E0, which I sometimes also call delta E0, this is equal to minus 2 multiplied by 96500 multiplied by 1.136, and this is about 219 kilojoules per mole. So under standard condition, oxidation of one molecule of NADH will produce so much free energy. This free energy is much larger than the hydrolysis of free energy of ATP, which under standard conditions is about 30 kilojoule per mole. So the energy from oxidation of NADH, which is about 219 kilojoules per mole, is used to produce several molecules of ATP in a series of steps, and this occurs in the mitochondria. So what really happens here, and I need to insert a picture of what occurs in the mitochondria. So the first step of oxidation of glucose, which is glycolysis, takes place outside the mitochondria, so inside the cell, and it produces pyruvate here. This pyruvate then enters the mitochondria. Now the mitochondria has two membranes. One is the outer mitochondrial membrane and the other is the inner mitochondrial membrane. The pyruvate enters the inside of the mitochondrion, which is also called the matrix, produces acetyl-CoA. You don't need to remember all the details, and this gets into the citric acid cycle, which produces a lot of NADH. NADH gets subsequently oxidized to NAD+, releases electrons which are used by the respiratory chain, used by a set of a few proteins sitting in the inner mitochondrial membrane to pump protons from inside the matrix to the intermembrane space in the mitochondria. And then these protons can be subsequently used to generate ATP by an enzyme called the ATP synthase. These protons, this synthesis of ATP uses what is called a proton motive force, which arises from both an electrical potential across the inner membrane and a chemical potential of protons across the membrane. And we want to look at the energetics of these processes. Typically, you need about 3 to 4 protons to generate one molecule of ATP. But once this proton gradient is established, how much free energy will be released when a single proton goes from the intermembrane space into the matrix of the mitochondria? And let's try to calculate that. So this free energy arises from two contributions. One is a chemical gradient, so concentration gradient, and the second is an electrical gradient, because there is a transmembrane electrical potential across the mitochondrion, and the outside, the intermembrane space, is at a higher potential than the inside. So let us look at the chemical contribution first. If you look at the chemical potential difference between the inside and the outside. This is given by RT ln the activity of protons inside divided by the activity of protons outside. Here activities are nothing but concentrations, so we can write RT log of the proton concentration inside divided by the proton concentration outside. And since log A by B equals log A minus log B, I can write this as RT ln concentration inside minus ln concentration of protons outside. When I say inside, I mean the concentration inside the matrix, and when I say outside, it means the concentration of protons in the intermembrane space. If you look at the logarithm of the concentration of protons, you can also write this as minus ln 10. Now we have a logarithm to the base 10 concentration of proton. This is nothing but the, I'm sorry, there's no negative sign here. This is nothing but negative of the pH, and ln 10 is 2.303, so this becomes minus 2.303 multiplied by pH. So if I use this equation and I put it here, I get a delta mu equal to RT minus 2.303 pH in minus 2.303 pH out. Or the delta mu arising out of concentration or pH gradient is equal to minus 2.303 RT pH in minus pH out. Wonderful. So this is the contribution of the chemical gradient. You also gain energy or gain free energy when a proton goes from the intermembrane space inside the matrix, because the potential outside is higher than the potential inside, and this difference in free energy is given by this equation. For the case of proton, this is equal to 1. So the total chemical potential difference is equal to the chemical potential difference arising out of pH gradients or concentration gradients and the chemical potential arising out of electrical gradients. And this is equal to minus 2.303 RT delta, the change difference in the pH inside and outside, plus Faraday's constant multiplied by the electrical potential difference. Now it turns out that this delta pH is about 0.5, and the delta E, which is the electrical potential difference, is equal to minus 0.15 volt. So the inside is negative with respect to the outside. The concentration of protons is higher on the outside, so the pH is lower here and the pH is higher here. That's why delta pH, which is pH in minus pH out, is a positive value. If you plug in these numbers, then the total free energy, or the total that you gain per mole of protons that is used up by ATP synthase and is moved inside the intermembrane, moves inside the matrix from the intermembrane space, is equal to minus 17.5 kilojoules per mole. So 1 mole of the transport of proton produces so much free energy. You can compare this with the free energy of ATP synthesis, which is about 31.4 kilojoules per mole. So a minimum of 2 protons, which will supply 35 kilojoules per mole, are needed to synthesize at least 1 molecule of ATP. But of course, this necessitates that the machines we are using are perfect and there are no losses. But because of inadequate coupling between the different components of this ATP synthase machine and other inefficiencies. Typically, we need about 4 protons for 1 molecule of ATP that is produced in the mitochondrion. So this is a little bit about the energetics of cells and how NAD plus and FAD plus are used to produce reducing power in the cell, which is eventually taken up in the mitochondrion to produce ATP molecules. It turns out that a single glucose molecule, the oxidation of a single glucose molecule, produces 38 ATP molecules in total, of which most are produced from NADH. So the oxidation of a single molecule of NADH produces about 3 ATP, which is about 100 kilojoules per mole. You can compare this to the free energy of NAD oxidation, which is about 219 kilojoules per mole. Yeah.